This lecture is about color. What is color? How does the human brain create color through the light that comes to the eye? What does color mean to us? What did color mean to visual artists like Turner? I'm Anya Hurlbert, Professor of Visual Neuroscience at Newcastle University.
So good evening and welcome to the second of the 2019 Darwin College Lectures on the theme of vision. Last week, in opening the series, Paul Fletcher spoke on visions and addressed such matters as the extent to which the model our brain makes of what is around us is factually correct and relates to reality, or perhaps doesn't, and how alternative realities can arise. Now, today, 25th of January, in case you've forgotten, it's Burns Night. It's an evening for haggis, tatties, bashed neeps, poetry, and a ceilidh. But tonight, here in this lecture hall, we are going to focus on colour. My love is like a red, red rose. So colour changes our vision. By colour, we see red roses and imagine love. But in another context, we might imagine red rage. By colour, we'd recognize those blue with cold, or perhaps we sing the blues. Now, those of us as old as me, able to remember black and white TV, will recall the transformation that colour brought. Black and white is kind of simply on-off, there, not there positioning. It makes shapes and shades. But colour brings a completely new dimension and a much deeper insight into what we see. Through photosynthesis, blue and red support all life. Green is actually effectively the throwaway waste, but for us, green symbolizes nature. So, colour is metaphor, it sends a message. Colour identifies political parties. You just have to watch the television and you see Jeremy Corbyn and Donald Trump sharing a great love for red ties to proclaim their allegiances. I don't know whether Nancy Pelosi and Theresa May both wear blue scarves. You can tell me. Where there's no colour, we get the message too. To show rage against evil, Picasso, that master of colour, discarded colour in his masterpiece, Guernica. Deepest of all, with colour, is the metaphor of hope. It was Archbishop Desmond Tutu. He reminded us of that. Red, orange, yellow, green, blue, indigo, and violet. The promise to Noah, the rainbow. Now, tonight, we welcome Professor Anya Hurlbert, Professor of Visual Neuroscience at Newcastle University, to talk about how our brains create colour, why it's subjective, why it's such an emotive aspect of appreciation and art. Fascinatingly, Anya has published on a wonderfully wide range of topics, from averse learning in the praying mantis. I wonder, does the doomed male get to learn from his mistake? To even measuring moisture in dried apples. But tonight, her focus is on colour and vision. Welcome, Anya. Okay. Thank you.
Thank you very much, Mary, for that wonderful introduction. I rather feel you should be giving this lecture instead with those insights. When Turner daubed a red buoy on his seascape Helvoetsluys, What did he mean? For Turner, the red created contrast, and in making that mark, he meant to generate salience and arouse entrance, to dominate his rivals and draw in his admirers. He painted in that red blob when the painting was already hung, and it was a response to Constable's opening of Waterloo Bridge, which hung opposite it in its golden glory. C.R. Leslie said at the time that the intensity of Turner's red lead, made more vivid by the coolness of his picture, caused even the vermilion and lake of Constable to look weak. The abundant yellows and blues and the evolution of color in Turner's works, as in this, The Fighting Temporary, voted Britain's favorite painting, painted when Turner was 63. It's a prime example of the rich, evocative, abstract before his time paintings that Turner, the pioneer, made. Illuminate several principles of color, even if he didn't understand them as we do now, as well as, most importantly, the meaning of color. One may say that he transformed from a painter obsessed with light and shade, seen here in an early engraving of the Falls of Clyde, to one consumed by color, as he transmogrified from the son of a hard-working barber from a troubled family to a wealthy, famous, and revered artist. This evolution is apparent in his treatment of favorite sites, which he drew from for his book of engravings, Liber Studiorum. He painted these sites again and again as he aged, adding color and removing line. Norham Castle in Northumberland is a prime example. Here you see a sketch of Norham Castle in his North of England sketchbook from 1798, all lines. Here, the mezzotint that appeared in the Liber Studiorum. Here, a watercolor of the same site in 1823. He's adding color, removing line as he evolves. And after a long period of introspection, as described by James Hamilton, in which he reminded himself of early subjects from the Liber Studiorum, he returned again to Norham Castle and created this painting, which is described as nowhere on this planet, just a blue rock and an unearthly cow. This is Norham Castle's sunrise around 1840. So his evolution from line and form, light and shade, to color and light reflects the debate between disegno and colore in art history. And that, in turn, mirrors the divide between form and color in the scientific study of visual perception. As Leonardo said, which is of greater importance, that form should abound in beautiful colors or display high relief? Leonardo came down on the side of relief, on form. And in his Burlington House cartoon here, he illustrates the power of form given through luminance variations only. In contrast to Leonardo's vision of form, Titian deployed color to illustrate the richness of the silk in which this man is dressed. This 16th century opposition between disegno and colore, form and color, carried on into the 18th and 19th century, and the French academicians thought color was too base, too sensual, too mutable to deliver deep truths. And there was a move away from statements about the wealth of pigments and the symbolism of color to a preeminence of form, line, and shape. As Ingres said, a noble contour may atone for many deficiencies. Delacroix, on the other hand, leading the charge for color and reveling in the exuberance of color, retorted, Ingres has not the slightest inkling that everything in nature is reflection and that color is essentially an interplay of reflections. Likewise, in vision science, some have dismissed color entirely, as Matthew Alpern, an American physiologist, did. Theories of visual object recognition put form as a primary cue. We can read Einstein from line orientation alone. We can recognize line drawings as fast as we can recognize color photographs, especially when we're trying to discriminate elephants from coffee cups. But there is evidence that color does play a role in object recognition, particularly in terms of recognizing and classifying diagnostically colored scenes, or object. Color speeds things up. People are much slower at recognizing abnormally colored bananas and abnormally colored forest and beach scenes. Color does help in other ways. Color helps to segment complex images into distinct materials. It helps to distinguish specular highlights from flecks of paint. It helps to distinguish shadow boundaries from material boundaries. In this picture of a fruit market, the cues conveyed by luminance, form, edges, are mixed. There's a mixture of shadow and bright sunlight. And if you look at the luminance image alone, what you see is this mixture of object boundaries, shadow edges, and sunlight edges. The chromatic image, on the other hand, taking away all the luminance information and instead leaving just the chromatic information, as Turner did with his latest Norham Castle, enables us to discriminate individual objects and see the difference between them. This idea underpins certain computer algorithms for automatically segmenting images into distinct areas so that they can then be recognized as different objects. And Tommy Poggio and I, back in 1990, proposed a segmentation algorithm which was based on hue variations only and did a better job than luminance edges in differentiating objects from each other. These ideas have been carried on by much greater computer scientists so that we can now do things like demarcate tigers on the basis purely of hue differences between the tiger's fur and the background. In providing a cue to visual segmentation, color also speeds up visual search, enabling us to find the red T's quickly in a group of blue T's, enabling us to find that blue book on our shelf or our rental car in a car park. Color also not only segments images into different objects, it tells us what those objects are made from. It tells us what material objects are made from, enables us to decide whether a particular fruit is edible. It enables us to recognize objects, to recognize one's red coffee cup and discriminate it from somebody else's blue coffee cup. So color in life and in vision science does many things for us. It helps us find objects, recognize them, classify them, but also it plays a strong role in our emotional responses to visual images. And for Turner, my argument is that he turned to color in order to express emotion, not just to represent a purer form of reality, but also to capture light. And his series of color beginnings, shown here, did just that, capturing emotion, as well as, in this case, daylight, literally laid out on the canvas. Carl Jung said, "Colors are the mother tongue of the subconscious." Indeed, colors do seem to have a direct route into the emotions, but why they should have such emotional heft is still a matter of scientific debate. As a vision scientist, I take the line that it's the biological meaning of color which leads to its emotional heft. This is an idea that Nicholas Humphrey here in Cambridge, by the way, first convincingly and beautifully expressed. So as a biological signal, as I said, color can tell us whether, for example, fruit is edible. Color can also reveal whether skin is healthy, and therefore it can supply information about the viability of a mate, for example. This gentleman being more attractive than this one with his sickly green glow. Color can signal the toxicity of a toadstool. Red is significant, and Turner knew that. In nature, red can attract or repel, it can signal toxicity or ripeness, anger, ruddy health, or sexual readiness. And we color scientists think of color as acquiring its meaning from these natural sources. So we naturally find red stimulating, arousing, attention-grabbing, because in evolution, red has signaled either danger or delight. So because color pierces so readily into the limbic system, we humans have also naturally learned to release color from form and then to apply it to other objects in order to signal their identity or their properties, to use color as a label and a symbol. So, for example, in man-made objects, color can symbolize group membership, as Mary said. Here, these aren't political parties, these are gangs in South Central Los Angeles in the 1990s, and color could really signal danger here. The Crips wore blue and the Bloods wore red, And as Aguilis Shirels said, you had to be really careful. I mean, you had to be very careful about what color you wore when you were in which neighborhood, your shoestrings, your belts. You could literally lose your life for wearing the wrong color in the wrong place. And it did happen. His friend was sawed in half. And in paintings, color can serve both as a symbol for good or bad, heaven or hell. And it might be that the pigment itself can help make that meaning and color. Here, in Sassoferrato's The Virgin in Prayer, it's all about the expanse of this expensive blue pigment, lapis lazuli, ultramarine, a color not seen in natural surfaces of objects. And when we thrill to the brilliant blue in this painting now, is this an emotion that we've learned from the pigment, whose blue has come to mean purity, heavenliness, and celestial richness, exactly because of its earthly origins in these very distant, arduously reached mountain mines? So we've seen what color can do for us in life and a little bit in art. We'll see more in a moment. But to make real headway in terms of understanding why color has aroused such deep interest from artists, philosophers, psychologists, we need to look more at what color actually is. Color is not a physical property of objects. Instead, it is a perceptual property made in the mind. There's no green dot in this image, but if you stare at the central cross and continue staring at the central cross, you will see a moving green dot in the periphery replacing the pink dots as they rotate around the center. The green dot is not there, but it's made by active processes in your brain. This idea that seeing is subjective and probabilistic, inferential, that visual images are ambiguous, has a long history in vision science and psychology, from Democritus to Locke, Goethe, and Newton. Yet we daily experience color as an inherent property of objects. We see the apples as red here, the cabbages as green. We think, we believe, we feel that the objects possess those colors. Thus, there is a dichotomy in our experience of color. Its inherent subjectivity in our experience of it, and the objectivity of color as it appears to be a property of objects in the natural world. The folk experience of color is that objects are intrinsically colored. And as Helmholtz noted, this is a necessary phenomenon for color to be a reliable indicator of material property. Ewald Hering, von Helmholtz's opponent in some ways, Hering was brilliant and irascible but felt that he wasn't acknowledged enough by his rivals, noted that color could only be a reliable indicator of object properties if we had the phenomenon of color constancy. He noted that if the colors of objects continually changed as the illumination changed on them, then we could never perceive constant colors of objects, and we could never see colors as fixed material properties of objects. If, as the light shining on the carrot causes the carrot to continually change its appearance, we would never be able to think of carrots as having the essential property of color. So what is this phenomenon that I've just referred to, color constancy? It is the bedrock of color perception. It is what gives color both its subjectivity and its objectivity. Color constancy is that phenomenon which enables us to perceive this painting by von Holzdunk as a painting of yellow lemons. Here the painting is illuminated by blue light, and we see the lemons as yellow. In this image, the painting has been illuminated by white light, and we still see the lemons as yellow. But if you compare the two images side by side, one illuminated by blue light and the other by yellow, you see that the difference in the light reflected to the eyes, because of the difference in illumination on them, is sufficient to change that central lemon into a lime. And this is the lemon seen in the context of the white-lit painting. It's unusual for an individual to switch off color constancy. It's so deeply embedded in the way our visual system works. But you could argue that Monet was able to switch off his color constancy. In his series paintings, the he was able to capture the color of the light, bypassing the mechanisms of color constancy, which should have kept the colors in the image stable. And he captured the radical changes in illumination as the day changed on this, in this case, Rouen Cathedral. He effectively disabled color constancy. And it's very unusual to see failures of color constancy in the natural world. But I would argue that you see a failure of color constancy in the phenomenon of Ayers Rock at sunset. In the center of Australia, Ayers Rock, also known as Uluru, is known as a rather magical place, partly for its dramatic changes in color. This series of pictures I took at sunset in Australia over a period of about 5 minutes, and as the sun changes its angle on the rock, it appears to change color radically from an orange to almost a deep purple. What does color constancy require in terms of the neural processes that carry it out in our brains? We could frame color constancy as a computational problem. What we are doing in seeing stable color appearance of objects under changing illumination is recovering something like the invariant reflectance properties of the object. All objects have constant properties, which mean that they reflect different amounts of the spectrum in different regions. So they reflect, the green leaf here reflects more light in the middle wavelength region. And that reflectance property, its tendency to reflect more light incident on it in that region, is something that remains constant. So the light reflected from the object is a product of the light source spectrum and the object's reflectance, the fraction of light it reflects at each wavelength. So the reflected light power follows this particular shape under this light source spectrum. The eye captures the light reflected from the object in 3 bands corresponding to the sensitivities of the 3 receptor types in the back of the eye, which we'll come back to in a moment, and records those responses as a triplet of 3 numbers. When the light shining on the object changes, the reflected light from the object will also change, even though its surface reflectance has not changed. So the cone responses, the receptor responses in the eye change. What the visual system must do then is take this changing set of receptoral responses and map them onto a constant set of responses under these changes in light. And this is not an easy problem computationally. Framed as a computer vision scientist trying to make a camera see constant colors, it's a very difficult problem which has intrigued computer vision scientists for decades. Back in the 1980s at MIT, this sort of problem was framed as an inverse optics problem. How do we start from a 2-dimensional image in which much information has been lost and recover a description of the 3-dimensional world? Similarly, in color constancy, how do we start with the light signal on the eye and disentangle the properties of surface reflectance from the properties of the illumination spectrum? That's an ill-posed problem. There isn't enough information to solve that analytically or directly. We can turn to artists, because artists have effectively been the most supreme, masterful inverse opticians around, in the sense that they know what to put into a 2-dimensional image in order to elicit the creation of the 3-dimensional world. So contemporary vision scientists think of seeing as neither passive, perfect, nor complete rendering of the outside world, but constructive, inferential, hypothetical. Although in the case of color, clearly the processes of color perception are designed to make our perceptions as close as possible, as meaningful as possible with respect to the physical properties. And because individuals differ, the experience of color perception that individuals create varies between individuals, because there are multiple ways to solve this ill-posed problem. For color constancy, these ways exist on different levels, and we know that each of these different processes contribute different amounts to the ultimate experience of constant colors under changing illumination. Chromatic adaptation in the eye, the adjustment of the sensitivity of the receptors to changes in the overall intensity of light contributes, as does, for example, memory color on a much higher level, on a cognitive level. And without going through all the different possible contributions of different perceptual mechanisms to color constancy, for which, unfortunately, in this lecture, I don't have time, what I'd like to do is just illustrate two of these, the phenomenon of chromatic adaptation and memory color. And we go back to our brilliant Ewald Hering to focus on memory color. He defined memory color as the color in which we have most consistently seen an external object. This color then becomes a fixed property of the memory image, and the memory colors in turn may further influence what we see on an instant-to-instant basis. And of course, because each individual has had a different life experience, memory colors might vary between individuals, and therefore the experience of color influenced by that varying memory color might be different for each individual. Memory color is what tells us that there's something wrong with this strawberry, and yet this is an acceptable apple. So memory colors are there, they're real, they are perceptual and mental phenomena. And what I want to try to do with this series of images is demonstrate to you the dual processes of chromatic adaptation and memory color in creating the colors of Dunstanburgh Castle on the Northumberland coast, another of Turner's favorite sites. This is a black and white image of the castle. How many of you have actually visited Dunstanburgh Castle? Oh, wonderful. A number of you. What I'd like to do now is just ask you to stare at the central black dot of this pseudo-color image of Dunstanburgh Castle. And if you continue to stare at that black dot, what we're doing now is creating chromatic adaptation in the receptors and the visual neurons in your higher visual pathway beyond the eye. And you continue staring at the black dot as I'm talking. This adaptation process will take place confined to different locations of the image. So the adaptation, the chromatic adaptation differs across the different locations in the image. And continue staring at the black dot, please. It is important that you keep staring at it and don't blink. And now you should see a color image of Dunstanburgh Castle. Through the processes of chromatic adaptation, which created an after image, but also because memory color, your memory color for sky and grass, has boosted the effects of chromatic adaptation. This is the full color image of Dunstanburgh Castle. And this is one of Turner's many paintings of Dunstanburgh Castle. He painted it from memory, and he clearly forgot where the sea was because he's moved it rather inland here. But of course, so as Hering said, memory color varies between individuals because of differences in life experience. And this illustrates the fact that at the heart of color, color is subjective. Colors are therefore one's own personal perceptual phenomena and possessions. And this fact explains, in part, the pettiness of some color scientists, I have to say. But nowhere more striking in recent history has the subjectivity of color perception been illustrated than in this phenomenon, which I hope some of you will remember having seen.
Is, I'm going to turn to something that's got the world talking. That dress.
That fucking dress.
I apologize.
It's blue.
I lost friends over this dress.
Oh my God.
This single photograph set the internet alight with a burning question.
Everyone was saying, is it black and blue or white and
Gold.
gold?
Yeah.
It's black and blue.
Is this dress blue and black or white and gold?
White and gold.
It's fucking awful.
I apologize for the language, but it does drive the point home. And I will come back to the dress later in the lecture. This was Gogglebox's take on a BBC show in which we demonstrated a phenomenon with the actual dress, which I'll come back to at the end. But the vehemence with which people clung to their dress color names shows how deeply people feel that color is an integral part of objects. And I'll argue they feel that way because of color constancy. So the consensus amongst vision scientists is that the explanation for the differences in the way people see that dress is due to differences in the way their color constancy operates. And I would just like to ask those of you who saw the dress for the very first time you saw it, how many of you saw it as white and gold? And how many of you saw it as black and blue? It's about half and half, just to illustrate that. So we'll come back to the dress at the end. But as I said, the dress illustrates this phenomenon that for ambiguous images, and in this case, the image was ambiguous because the sources of light were ambiguous. We didn't know the light source color. In fact, there were probably two sources of light on it. We didn't know the dress color to begin with. There was a great deal of ambiguity in the image, and individuals had to work with different constraints and assumptions in order to recover the apparent constant color of the dress. But to go a little more deeply into what color constancy is and how we measure it and what influence it has on the colors we see, let's look a little more closely at the fundamentals of color processing.
Light is reflected from the world around us into the eye. At the back of the eye is the retina, a thin layer containing the light sensors called cones. There are 3 different cone types, red, green, and blue. Each one is stimulated by different wavelengths of light. The cones reduce the full visible spectrum to 3 values at each location on the retina. After more processing, the cone's signals are sent via the optic nerve to the brain, where the colors we see are finally created.
That was the voice of Lalla Ward, who made this film for us, which we showed at an exhibition at the National Gallery, which I'll talk about in a moment. So at the back of the eye, the 3 cone types are arranged in a cone mosaic which differs radically between individuals. And this is just one of the striking individual differences in the sensory apparatus of color vision, but it doesn't explain the striking individual differences in color perception. This trichromatic visual system that we have evolved from a primordial system. 100 million years ago, our ancestors possessed a primitive dichromatic color vision system which enabled discrimination between short and long wavelengths only. And this was enabled the system to build cells which opposed the blue cone system against the output of the middle to long wavelength system, and created a space in which blue was opposed against yellow. And this system would be ideal for following the colors of daylight, because daylight is a mixture of sunlight and skylight, and it varies with time of day, season, and geographical location. Its chromaticity varies along a line close to the Planckian locus of black body radiation in the color diagram. And this Planckian locus, along which the chromaticities, colors of daylight vary, lies very close to the line that isolates the responses of this short wavelength cone. So it seems that this short wavelength cone system would be ideal for following the changes in the color of daylight. And it was much later in evolution, as eloquently formulated by Professor John Mullen of this university, that our modern trichromatic system evolved via a split in this long wavelength cone into 2, a middle wavelength cone and a long wavelength cone. And this was about 30 million years ago. And this enabled the formation of a second chromatic channel, which we, in a rather poorly named term, called the red-green channel, in which the output of the long wavelength cones are pitted against the output of the middle wavelength cones, forming a 2-dimensional chromatic space in which the axes represent these two channels, the red-green channel and the blue-yellow channel. There are two hypotheses that have been put forward for why this split in the LM cone pigment came about. One is that it was driven by our need to discriminate red berries against green leaves. This is the frugivory and folivory hypothesis put forward by Sumner and Mullen. And another is that because we became bare-skin animals about that time, that it was driven by our need to read emotional signals caused by slight changes in blood oxygenation in the face. One possibility is that frugivory drove the evolution, but then our ability to read social sexual signals from face color changing piggybacked on that change. And artists, of course, have also capitalized on this ability of ours to read signals in the flushing of the cheek. So we could say, therefore, that we have two systems, a blue-yellow system, which is optimized for detecting changes in light, such as Turner captured in his color beginnings images of daylight, and a red-green system designed to enable us to detect and recognize object types. And there's also some evidence that the color neurons in primary visual cortex are actually tuned to the colors of daylight, which don't fall exactly on the S-cone isolating axis. It's also the case that an analysis of the chromaticities of natural surfaces show that they fall in a particular quadrant of red-green cone contrast space, confirming that it's the red-green axis which best characterizes differences between objects. And note that most objects fall in a sort of warm color of color space as opposed to the cool colors of the sky. These blue, yellow, and red-green opponencies inherent in the neural processing of color were, of course, naturally, observed and plumbed by visual artists for all the vigor and resonance they could yield in visual art long before these opponencies were understood and described by vision scientists. Artists were guided by color circles like this developed by Newton, who was the first to arrange hues in a systematic arrangement in which colors were complementary to each other. Here he opposed red against blue, yellow against violet, and green against purple. Moses Harris, in his natural system of colors, proposed a different opposition, maintaining the primary position of red, blue, and yellow, and opposing those against green, orange, and purple. Goethe then proposed a different color circle, but he was much more obsessed with the polarity between light and dark. And it was Goethe's circle that inspired Hering to develop his theory of opponent colors, red versus green and blue versus yellow, which we now recognize as the basis for the physiological cone opponent space, which I described. The fact is that the scientific definitions of which colors are complementary to each other and how they are defined, for example, as Chevreul defined them as the pairs of colors which heightened the greatest dissimilarity between them when they were juxtaposed. The fact is that those definitions are still not set, and between scientists there's about as many definitions of complementary colors as there have been uses of complementary colors by artists. Van Gogh here, for example, used a very jarring dissimilar combination of colors, reds, greens, yellows, all very saturated, expressly to achieve this violent clash. He described this painting as one of the ugliest I have done. Turner, himself, taking up Goethe's emphasis on light-dark polarity, yet rejecting the notions of other color circles, devised his own color circle. This wasn't a hue circle proper. It was more like a pyramid in which yellow reigned supreme at the top. He basically flattened Harris's circle. He said, sink the yellow until it light into red and blue, and hence two only, light and shadow, day and night, or gradation of light and dark. In homage to Goethe, Turner created this famous pairing of paintings late in his life. He expressly uses color, both chromaticity and luminance, but almost only these cues, no form again, to illustrate the morning after the deluge and the evening of the deluge. The morning is a brilliant cyclone of colors converging on yellow and white in the center, and the evening, blackness circling a gray-blue core. And these two paintings really capture the color of light and also signal the time of day. And they partly inspired us, my colleagues and I at the University of Delft, to look into how good people are at reading the time of day from the color of light. I'd like to just take a minute or two as an aside to talk about this. So color, the daylight varies in both color and lux, overall power during the day. And these are measurements that Naomi Gross made in Northumberland recently. And as you see, at dawn, the light becomes more blue while it's still very dark, and then it becomes yellower as the lux levels increase. And then the chromaticity, the color of light, stays roughly the same for the rest of the day, but the lux rises steeply and then falls again. And then at dusk, symmetrically to dawn, the blueness increases and then drops again. So we could read time of day, in theory, from the chromaticity and lux changes in daylight. Have other painters than Turner exploited this regularity? We asked this question using an MTurk experiment online. We simply gave people 100 paintings and asked them to rate each painting in terms of what time of day it depicted. They had to just choose the time of day from 7 choices. As I said, I did this with colleagues at Delft University. So 104 paintings. And the punchline is that people were very consistent in rating the time of day in these paintings. The time of days varied across the paintings, but with good agreement between individual participants. And they varied significantly with paintings. So percentages ranged from 84% morning to 6% morning. I'll just show you a few examples. So this painting was rated as, anyway, this painting was rated as, 69% rated this painting as noon or afternoon. 65% of people rated this painting as evening. And 77% of people rated this painting as night. So what were people responding to in making these time of day judgments? It turns out that there is a relationship between the chromaticity and luminance of the painting overall and the rating of time of day. Dark blue paintings were rated as being in the evening, and bright yellow paintings as being more likely in the morning. So this plot just shows the mean time of day rating going from morning to night as a function of color of the painting, which we call correlated color temperature here. And this shows the mean time of day rating as a function of the mean image luminance. So the brighter the image, the more likely it was to be rated in the morning. So it seems that people and painters embody an understanding of the regularity of changes in the color and lux levels of daylight. Some do so very explicitly, as Corot did with these paintings, but painting the same scene at different times of day and labeling them with the time of day. So so color and lux can indicate time of day by capturing the changes in the color of daylight. Is this not a violation of color constancy? If we can see the color of light, are we not then seeing constant colors of objects? Before getting to this question about color constancy and how it depends on our ability or inability to see changes in the color of light, I did just want to mention, take just 2 minutes to talk about the other ways in which we respond to light, because I can't really give a lecture on color and vision and what goes on in the eye and brain without mentioning this radical discovery that was made 17 years ago. Rods and cones are not the only light sensors in the eye, because you can create mice who don't have any rods and cones, but they can respond to light. So this means that there's another light receptor in the eye, and it turns out this light receptor is in the intrinsically photosensitive retinal ganglion cells, which is in a different layer from the classical photoreceptors, the cones. And this set of IPRGCs projects to a different part of the brain, a non-visual part of the brain, in particular the suprachiasmatic nucleus. And the situation is so much more complicated that I first implied with my tidy picture of two chromatic channels. And I won't go through this in detail, but just to say that these IPRGCs that contain this pigment that responds directly to light are also fed by short wavelength cones via an inhibitory link. And these cells project not only to the visual pathway, but also to this non-visual pathway, the suprachiasmatic nucleus. So there's considerable crosstalk between the two pathways via cells that are responding to the short wavelength signal and responding to the color of light. And the sensitivity of this pigment in the IPRGCs falls between the sensitivity of the short wavelength cones and the red and green cones. So it is definitely, well, definitely, it's thought, going to be contributing to our color perception too, via these crosstalk mechanisms. These IPRGCs, together with the short wavelength fed cells, are optimally designed to detect the bluing of light pre-dawn and the yellowing of light at dawn as lux levels increase. So that signal conveyed by the IPRGCs is really an awakening signal. And the fact that these cells feed directly to the suprachiasmatic nucleus enables them to govern our behavior in response to the changes in light during the day. So the human response to light is a dual response. We have a conscious visual response and an unconscious response which is entrained to the changes of light during the day and therefore entrains our circadian rhythms, affects our arousal and our mood. So these are two pathways which are mediated by the classical photoreceptors in the eye and the intrinsically photosensitive retinal ganglion cells. And we have been studying the interaction between these two pathways using special lights which we can modulate in different regions of the spectrum so that we can separately modulate the sensitivity of the melanopsin-containing intrinsically photosensitive retinal ganglion cells and the classical receptoral pathways. And with these lights, for example, we've shown that with different spectral variations in light, we can affect the release of melatonin, we can affect sleepiness, cognition, and mood. And I did say I wanted to just spend 1 minute on this. I'm really racing through this, but I just wanted to give you the take-home message, which is that blue and white light at night equally suppress melatonin and suppress sleepiness, but amber light does not suppress melatonin, even though it's very bright, and allows sleepiness to be resumed at night. So the take-home message from this, and I'm not going into the experimental details, and I apologize for zooming through it, is that at night, if you want to see and perform better and not affect your natural sleepiness, use amber light, not bright blue-enriched white light or blue light alone. So we can ask the question, given that we have color constancy and we know that the visual system has evolved under natural daylight, is our color constancy optimized for spectral changes in daylight? To ask this question, we need to have a way of measuring color constancy. And there are multiple ways to measure color constancy. The classic ones being to match surface colors under two different illuminations. Other methods being to match the light color or to detect a change in illumination. And I want to talk about these two methods very briefly to illustrate that we do have a bias for color constancy under daylight. So measures of surface color constancy using stimuli like this, asking people to match the colors of two surfaces in scenes simulated under different illuminations, suggests that color constancy is actually not very good. Estimates range from 10% to 85%. But again, we can ask the question, is color constancy better under some illumination changes than others? For example, here we see a scene illuminated by light changing over the course of the day from dawn to dusk. And we're rather insensitive to these changes in light. The colors of objects appears to stay the same, although the actual light coming from the scene is radically different, as you can see if I play each of these at different, if I choose snapshots from the film at different times. So this suggests that we might be relatively impervious to changes in the color of daylight if it occurs at the same time scale as typical during a natural day. So we ask the question, how good are we at discriminating changes in illumination in a scene? And I want to just ask you to do this little experiment. What the experiment involves is looking at a scene and then comparing it to two scenes following, and just say whether the illumination has changed or not under this, in the interval between them. And we perform this experiment in the lab, in our light room, where we have what we call a Mondrian box, illuminated by these tunable LED light sources, whose illumination we can sculpt in real time. And I'll just give you an example of one trial. The observer is looking into the box at the Mondrian and is asked to view a target scene and then say whether the first or the second scene has the same illumination. So, ready, steady. First scene, 1 or 2. Which best matched the target? The second, exactly. So that target had the same illumination. So by varying the change in illumination between the scenes in different amounts and along different directions in color space, we can determine how far we can push the change in illumination for, until the person can just see it. So how much can the illumination change subliminally, maintaining constant color appearance of the scene? And we examine directions in color space which fall along the daylight locus and then fall orthogonal to that along a red-green locus. And what we find is that people are much better at seeing changes in daylight if these changes fall along the red or the green direction, which are more unnatural directions for illumination color change. The accuracy is worse for a change along the bluish direction, and this is whether we use a gray background or a Mondrian background. And although yellow also lies along the daylight locus, people are a little better at discriminating yellow changes from blue changes than compared to blue changes. So we call this a blue bias for color constancy. Color constancy is better. The scene colors stay more constant if the illumination is moved in the bluish direction. And this is a very robust finding, which we and our colleagues have now shown in multiple situations. And we describe this as a discrimination ellipse in color space where the threshold is much bigger for seeing changes in the bluish direction. And one of the experiments where we've shown this blue bias was in a public experiment at the installation in the National Gallery that I referred to. So I'll just take you through that experiment where instead of in the lab where we could test 8 people with multiple trials, in the National Gallery installation, we could get very reliable responses from 1,800 people, although actually 10,000 people took part. And this was at the summer exhibition Making Color in 2014 at the National Gallery, where people sat in the cinema after viewing a wonderful exhibition on the physical process of color, the pigments in color. They sat in a cinema and viewed this reproduction of a Gauguin painting, which was placed on the wall next to a color print of the same reproduction. And the film I showed you about the receptors at the back of the eye was projected on these screens, which were turned off when the people did the experiments. Individuals did experiments and delivered responses to us by inputting their responses on little clickers, and we were therefore able to ask them the same question that I just asked you, which was...
In two lights, one after the other. You must choose which light matches the target light. Now, lights off. Which of these lights matches? Light number 1 or light number 2?
So again, we measured ability of people to detect the change in illumination, and again we found that accuracy for discriminating changes in illumination was worse along the blue direction. And this again suggests that color constancy is better for bluer illuminations, and it also enables us to cross-validate from the laboratory to real-world viewings that the blue bias exists, and it might feed into different ways to illuminate paintings in galleries. I want to just describe briefly one more experiment, one more way we measure color constancy, which also leads to the same conclusion that we have a blue bias. But we get there by asking, what is white? Why do we ask what is white? I'll ask you first. How would you describe this color? White. I hope I'm answering. And white again. But each of these are, of course, different whites. It's the same scene on the same day at different times of day. And yet, at each moment, we see white snow-laden sky. White can take on any color depending on time of day and depending on the daylight. And it's this fact that is used in measurements of color constancy. The task we use is called achromatic adjustment. We ask people to adjust a surface to look white, and that thing which is white will reflect exactly the color of the light. So if the person adjusts a surface to be yellow under yellowish light, they are behaving as if perfectly color constant. They have adapted to the color of the light, because a white surface will reflect exactly the color of the light. So if a yellow surface looks white under yellow light, that's an expression of color constancy. And this is a technique which has been used by many people over many years, going back to a wonderful experiment by David Brainerd in 1998. But recently we've done the experiment again in the Lightroom, where people adjust the color of this surface, which in fact is a smartphone display, to look white under changing illumination. And we then measure the chromaticity that the individuals have adjusted the device to be. And I should just point out, of course, that painters know that there is no white in nature in itself. As Renoir said, white does not exist in nature. You have to admit you have a sky above that snow. Your sky is blue. That blue must show up in the snow. A white surface will reflect the color of the light. And what we find when people adjust the color of a surface to be white under different colors of lights, here represented as the open circles, their matches are not perfectly aligned with the actual color of the light, expressing imperfect color constancy. The black squares represent their matches, but for bluish lights, the errors are much smaller. This suggests a bluish bias. When the light is actually blue and the surface is reflecting bluish light, it looks more white. So there is a blue bias in achromatic settings. And when we test a greater range of lights with greater range of chromatic colors, we see the errors are larger, but they're all pointing towards the daylight locus. So there is a tendency to see daylight chromaticities as white, and more perfect color constancy when the light actually matches daylights. To get at what people are actually seeing when they're seeing white surfaces under colored lights, Jan Kuchera in my lab has recently done a set of experiments in which he had people view a piece of white paper shown here Again, this is in a box illuminated by tunable lights, and they have to match the color of that white paper with the color of a tablet. This is a view of a tablet also in the box. And the instructions appear on the tablet screen, and they have to make the color of the tablet screen match the color of the white paper under different colored illuminations. And what Jan has found in this case is that people's matches to the color of that white surface under different colored lights are very accurate. So the black square here indicates the color of the colored light, and all the crosses and dots indicate people's matches. So if the light is green, they match the white paper under green light to the color of the green light. So they're very accurate. But we also ask them to name the color of the white paper under the chromatic lights, and it's only when the paper is under bluish lights that they call it white. So white paper under very chromatic lights does not look white. Color constancy can be pushed beyond its limits if you make the light chromatic enough, unless it's blue. There is a blue bias in color constancy. And that brings us back to the dress.
Dr. Helen met a really clever professor who'd set up a science lab under a willow tree.
Professor Anya Hurlbert is a vision psychologist.
But that's the only time her job's actually come in handy, when that fucking dress went mental. Say, don't worry, I'm here. The vision psychologist's here.
I'm on the
Nobody
kit.
freak out.
So,
Dr. Helen.
vision scientists all over the world were suddenly very popular with the press. But we set up this experiment with the real dress and two lights.
Open your eyes.
Yellow and blue lights.
Blue, black. It's white with gold trim.
White and gold.
White and gold?
Yeah.
White and gold.
I think it's blue and black.
So even the real dress could be made to be seen as white and gold or blue and black by different people at the same time viewing it because of the ambiguity of the lighting. And we showed in separate measurements, as have many others, including Christoph Witzel, for example, that it is indeed the person's assumption about the illumination color which affects their conclusion about the colors of the dress. So those people who see the dress as white and gold tend to see the light, the illumination on the dress as being darker and bluer, and those people who see the dress as blue and black tend to perceive the illumination on the dress as brighter and yellower. So individuals differ in the assumptions they make about the illumination on the dress. They assume it's daylight, which is why this illusion works so well, because it is an array of blue and yellow chromaticities. They assume it's daylight, but they assume it's different types of daylight. And in fact, this is not the only blue-yellow dress around. Guzzoli, long ago, might have been trying to achieve the same effect with his striking contrast of blue and gold, or he might have been trying to display the properties of Changeux fabric. So blue is special, and we know that there's a blue bias in color constancy. There's also an emotional bias for blue. Many studies of color preference have shown that people prefer blues. This is average preference ratings collected by Karen Schloss and her colleagues, which show that people's preferences for blue are higher than their preferences for yellows and browns. And we likewise showed similar results, different population of people, different ages. Karen Schloss and Steve Palmer postulated that people have these preferences for colors because they associate them with objects with the same colors. So people like blue because blue is the color of clear water, blue skies. They dislike dirty browns and yellows because they're the color of excrement, puce green because it's the color of vomit, for example. So this is a deeply embedded notion. We have emotional responses to colors because they signal properties of objects, and we transfer those emotional responses from the objects to the colors themselves. And this blue being such a color, wonderful color of light, becomes even more unusually appealing when it's applied to a surface because it doesn't occur so readily in natural objects, which tend to be red and green. And Yves Klein, of course, illustrated this very dramatically by applying this deep, beautiful, brilliant blue to the female form. And finally, coming back to Turner, Turner loved blue because when he was a teenager, he said he would lie on his back, look at the skies, and then go home and paint the skies on canvases and sell them in the market. So the celestial blue of the sky became a real monetary asset. His red in Helvoetsluys thus carried meaning on multiple levels. The red target was designed to deliver contrast in a sea of blue. And his resistance to opponent color schemes, in fact, is slightly negated by the fact that he so brilliantly captured the opposition of blue and yellow-orange in the fundamental structure of daylight. This is more masterful, more evident in Turner's masterful renderings of light itself. So the red object that he threw into his sea of light was a brutal object intruding on a turbulent sea, but it was a weapon. As Constable said of that red mark, he has been here and fired a gun. I just want to thank all the people who've contributed to the experiments that I talked about. Thank you very much. Sorry, I overran. I'm so sorry.
Thank you. Thank you very much indeed. So Anya has reminded us that colour is a representation of particular frequencies of light, EM radiation, but that how we see any different given coloured object depends not just on our eyes and our brain, the object, but the object itself, the light source, what we think the light might be, perhaps the background. I mean, how can we trust our judgment of colour and our eyes at all? It's definitely something, to me, alarming, extraordinary, although clearly some of the time we can judge that a white piece of paper is white, but at other times we're quite sure that it's something else, and that we should be careful about what colours we are reading on, living on, late at night before we go to bed. We should have changeable light bulbs, tunable light bulbs. A whole range of questions comes to me. I'm sure conversation later this evening will go on and on and on. Next week, the subject of our lecture next week, the third in this series, is evolution and the eye. And you can see that it's going to follow on very well from this lecture this evening. So next week we have Dan Erik Nilsson, the professor of functional zoology at Lund University in Sweden, who's going to talk about the wide variety of different types of eyes in the animal kingdom, how they work and how they've evolved. So we hope to see you then. But for now, thank you, Anya, very much indeed for coming to talk to us this evening.